The main aim of Kelvin cycle is to produce glucose, which is the byproduct of photosynthesis. So C4 pathway actually occurs whenever the plants do not receive enough of the carbon dioxide. RUBP is ribulose 1,5-biphosphate. So photorespiration does not occur in the C4 plants. It is only seen in the C3 plants. Hello everyone, a warm welcome to another session on chapter 13, that is photosynthesis in higher plants. I am Dr. Divya, biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session, we had discussed about the light reaction of photosynthesis, and the light reaction occurs in the grana of the chloroplast. And under light reaction, we studied about two important light harvesting complexes, which was called as the photosystem 1 and the photosystem 2. So if photosystem 2 and 1 both are involved in the photosynthetic process or in the light reaction, then it is called as the Z scheme or the cyclic photophosphorylation. On the other hand, if only photosystem 1 is involved in the light reaction wherein the light harvesting complex is P700, then it is considered as a cyclic photophosphorylation. And we also learnt that the ATP, that is adenosine triphosphate, which supplies energy for the photophosphorylation, and the NADPH that actually helps in the transport of electrons throughout the photophosphorylation, both are produced in a process called as chemosmosis in the grana of the chloroplast or in the thylakoid of the chloroplast. So we also learnt about the chemosmosis process. In today's session, we shall concentrate on the mechanisms that are involved in the dark reaction, wherein we shall study about Kelvin cycle, which is also called as the C3 cycle, and we shall also study about the C4 pathway or the C4 cycle, which is also called as the Hatch and Slack cycle. So let us begin today's session. So Kelvin cycle. So Kelvin cycle is named based on the scientist who discovered it. So a scientist, his name is Melvin Kelvin. He was the one who actually discovered this Kelvin cycle, and the cycle is also called as C3 cycle. So why it is called as C3 cycle? The reason behind it is the first stable compound that is formed during the Kelvin cycle is a 3-carbon compound. It is a 3-phosphoglycerate or it is a 3-phosphoglyceric acid, which is a 3-carbon compound. And the name of the 3-carbon compound is 3-phosphoglycerate. So 3-phosphoglycerate is a 3-carbon compound, and it is the first compound that is actually formed during the Kelvin cycle. That is why it is also called as the C3 cycle, and the plants which undergo this Kelvin cycle or the C3 pathway are called as the C3 plants. And for easy understanding, the Kelvin cycle is divided into 3 steps. One is carboxylation, the other one is reduction, and the third one is regeneration. So we shall study in each of these steps what happens. So first talking about carboxylation. So when I say carboxylation, it means addition of carbon, right? So here, how will the plants get carbon? It is got from the atmosphere, right? So plants during the daytime, when they open their stomata, the gases are exchanged, wherein carbon dioxide that is present in the atmosphere is taken in by the plants. So carbon dioxide, and we know that carbon dioxide along with water, they are the raw materials which are very, very important for photosynthesis, right? So this carbon dioxide will be taken in. So 6 molecules of carbon dioxide. How much carbon dioxide? It is 6 molecules of carbon dioxide will be taken in. So these 6 molecules of carbon dioxide will be taken in by the plants, and for this one particular compound will help. It is called as the RUBP, that is RUBP, which is nothing but ribulose 1,5-biphosphate. And this ribulose 1,5-biphosphate is a 5-carbon compound. It is a 5-carbon compound. So what did we understand under carboxylation? The ribulose 1,5-biphosphate helps the plant to absorb the carbon dioxide, that is 6 molecules of carbon dioxide from the atmosphere. Therefore, this ribulose 1,5-biphosphate, if it has to convert the carbon dioxide or reduce carbon dioxide into a 3-carbon compound. So what happens here is 6 molecules of carbon dioxide are being absorbed with the help of ribulose 1,5-biphosphate, which is called as RUBP, and it is being reduced to a 6-carbon compound, right? So if it has to be reduced to a 6-carbon compound, there is one enzyme which helps. It is called as the Rubisco enzyme. So there is an enzyme which is called Rubisco enzyme. So this Rubisco enzyme actually helps in the splitting up of the carbon dioxide. Again, to make you understand, so here carbon dioxide is accepted, 6 molecules of carbon dioxide is taken in by the plant with the help of RUBP, that is ribulose 1,5-biphosphate, which is a 5-carbon compound, and this absorption of the carbon dioxide by the ribulose 1,5-biphosphate is helped by an enzyme called as Rubisco. Now, we have 6 molecules of 6-carbon compound inside the plant. 6 molecules of 6-carbon compound is there. So, this 6-carbon compound, actually it is quite unstable. So because it is unstable, what happens? Very soon it will split into 2 3-carbon compounds. So it is split into 2 3-carbon compound, which is called as 3-phosphoglycerate. So that is why, because the first compound that is formed in the Kelvin cycle is a 3-carbon compound, which is nothing but 3-phosphoglycerate. It is also called as the C3 cycle, and it is more stable when compared to the 6-carbon compound. So now this 3-phosphoglycerate, which is a 3-carbon compound, so here totally there are 12 molecules of 3-phosphoglycerate. Why? Because 6 into 2, it will become 12, right? So 6 molecules of the 6-carbon compound are split into 2 molecules of 3-carbon compounds, right? So therefore it will be totally how many? 12 molecules of these 3-phosphoglycerate will be formed, which is a 3-carbon compound. It is a 3-carbon compound, as I told you. So much is understood under carboxylation. So these 12 molecules of 3-phosphoglycerate that are there, they have to get reduced. Reduction has to occur, wherein they get reduced into a triose phosphate. So here, if reduction has to occur in the second step, here they'll make use of energy from ATP, which combines with NADPH to form ADP, and one inorganic phosphate gets added. So here what happens in the reduction process is, here it forms the addition of one phosphate group will take place, because phosphorylation is occurring here, because ATP is donating a phosphate group here. So here what happens? A phosphorylation will occur in the reduction process, and that will get converted into glucose, or which will later on be stored in the form of starch in the plants. So that is what happens in the reduction process. Next, in the third step, after reduction, there is regeneration. Why regeneration occurs? Because there are 6 molecules of carbon dioxide, or 6 molecules of the 6-carbon compound has been formed, right? So each of these 6 molecules of 6-carbon compound, if it has to get reduced, it should enter through 6 cycles. It has to pass through 6 cycles. So to make you understand again, first from the carbon dioxide will be taken in from the atmosphere with the help of a compound, with the help of a 5 carbon compound called as RUBP, that is ribulose 1,5 biphosphate, taking the help of an enzyme called as Rubisco. Now, what happens here is inside the plant there will be 6 molecules of a 6 carbon compound will be formed. So, this 6 molecules of 6 carbon compound, each of these compounds, they are quite unstable in nature. So, that is why they get converted into, or they get split into 2 molecules of 3 carbon compound, wherein at the end 12 molecules of 3 carbon compound, that is 12 molecules of 3-phosphoglycerate will be formed. So these 12 molecules of 3-phosphoglycerate will undergo a reduction process wherein it should get converted into glucose. So here the addition of phosphate group will take place with the help of ATP and NADPH, and it will get reduced to glucose, which is a triose phosphate. It will get reduced into a triose phosphate and therefore glucose is formed. So what happens here? One carbon molecule, one of the 3 carbon compound has undergone reduction to form glucose. Like this, how many are remaining? Again here there are 6 molecules of 6 carbon compounds are remaining, right? So each of these 6 molecules of 6 carbon compound, they have to get reduced, right? They have to split, they have to get reduced. So each time 12 will be formed. So they have to get reduced, they have to get, they have to split, they have to form 3 carbon compound, and then they have to undergo reduction and then form a glucose molecule. So each time they have to complete 6 cycles. So each time if they have to complete 6 cycles, what happens? Regeneration should occur here. What should get regenerated? RUBP should occur. Why? Because only if RUBP, that is ribulose 1,5 biphosphate is there, then only the further process can take place, right? So if cycle has to continue uninterrupted, every cycle, that is if 6 cycles has to complete, every cycle a new RUBP compound should be formed, which is a 5 carbon compound should be formed there, right? So in the third step, regeneration of the RUBP will occur. So if regeneration of RUBP has to occur, then each step 1 molecule of ATP will be utilized. So in each step, 1 ATP will be utilized for phosphorylation, wherein it will form ribulose 1,5 biphosphate and the cycle continues. So this is about Kelvin cycle. So Kelvin cycle, the main aim of Kelvin cycle is to produce glucose, that is, which is the byproduct of photosynthesis, right? So we had written an equation wherein 6 molecules of carbon dioxide combines with 12 molecules of water in the presence of sunlight and chlorophyll. They get converted into C6H12O6, which is glucose, wherein 6 molecules of oxygen is liberated and 6 molecules of water is also formed. So it was a simple step which we had written, but did we imagine that to form glucose, if plants had to make glucose, it has to undergo this particular Kelvin cycle wherein different enzymes are being involved, and therefore carbon dioxide and water here is one of the raw material wherein carbon dioxide will be utilized by, or it will be taken in by the RUBP, that is ribulose 1,5 biphosphate, which is a 5 carbon compound, by the help of an enzyme Rubisco, and the cycle will produce. So here therefore what happens, the 6 molecules of, or you can simply remember as the 6 carbon compound that was formed, it is very unstable, and therefore it will be split into 2 3 carbon compounds. Therefore, because the first compound that is formed is a 3 carbon compound which is 3-phosphoglycerate, it is also called as the C3 cycle. So at the end, in the C3 cycle, what gets produced? The glucose will get produced, and every cycle when it continues, the RUBP has to get regenerated with the use of 1 ATP molecule. So that is how the Kelvin cycle continues. So next, moving on to one more cycle, that is the C4 pathway. So C4 pathway actually occurs whenever the plants do not receive enough of the carbon dioxide. So say for example, sometimes because of environmental stresses. So during environmental stress, what happens? Say for example, there is a drought condition or the temperature is very hot. Because of that, the water availability is very less. In such cases, what happens? The plants have an adaptive feature, right? So what they do is they close this stomata even during the daytime. So why do they close the stomata even during the daytime? It is to prevent the excess loss of water from the plants, or to reduce the rate or completely stop the rate of transpiration, so that during the unavailability of water, whatever water is there inside the plant, that will remain as such, and therefore the plant can utilize that for the photosynthetic process. So that is the concept behind it. So here what happens sometimes is, in such unfavorable conditions, the availability of carbon dioxide to the plant will be very minimal, and the plants will have to utilize only that carbon dioxide which is present inside its cell. Say for example, the mesophyll cells, because in the epidermis, few of the cells, they get converted into the stomatal apertures or the stomatal openings through which the carbon dioxide enters. And once the carbon dioxide enters, what happens? It enters into the mesophyll cells. So below the epidermis of the plant is present the mesophyll cells, right? So these mesophyll cells, they trap the carbon dioxide and they will be utilized for various processes. So how much amount of carbon dioxide is there inside the mesophyll cell? Only so much can be utilized by the plant, because next what happens, the stomata would have closed due to unfavorable environmental conditions, and therefore the carbon dioxide will not be able to enter into the mesophyll cells of the leaves through the stomata because they are closed. So at such times what happens, they'll have to utilize only those carbon dioxide that is been taken in prior itself. So in such conditions, the plants undergo a pathway called as the C4 pathway. So we shall study what exactly happens in the C4 pathway. So what they have done here is, they have drawn the cells, enlarged structure of the cell, wherein this is the mesophyll cell. So the mesophyll cell and the bundle sheath cells. So bundle sheath cells usually help in fixing the carbon dioxide in the case of C4 plants. So what are C4 plants? Those plants which are subjected to a lot of stress, that is environmental stress. Therefore, the availability of carbon dioxide is very less in them. So usually bundle sheath cells are a group of cells or a mass of cells that are usually found in the vein regions or surrounding the vascular bundles of the leaf and all that. So these cells, in the case of C4 plants, they actually help in carbon dioxide fixation, and not just that, they usually form a separate compartment, and in that compartment the carbon dioxide fixation will take place. So here what happens in this particular case is, in the mesophyll cells, the atmospheric carbon dioxide would have already been taken in by the plant. So here the atmospheric carbon dioxide that is taken into the mesophyll cells by a compound called as phosphoenol pyruvate. So this phosphoenol pyruvate actually, it fixes the carbon dioxide, wherein it gets converted into a stable compound which is called as oxaloacetic acid. It is OAA, which is oxaloacetic acid. So this oxaloacetic acid is actually a C4 compound. It is a 4 carbon compound. So this 4 carbon compound, now that is stable. So remember, there in the case of Kelvin cycle, what happened? The carbon dioxide was taken in by which compound? That it was taken in by the RUBP, right, with the help of an enzyme Rubisco, and that 6 carbon compound which was unstable was converted into a 3 carbon compound, that is a 3-phosphoglycerate, right? And that was stable, and that 3-phosphoglycerate later on was utilized in the Kelvin cycle. But in this case, what happens in the case of Sheath pathway, the atmospheric carbon dioxide will be assimilated by a compound that is phosphoenol pyruvate. This phosphoenol pyruvate fixes the carbon dioxide compound and makes it and converts it into a stable 4-carbon compound, which is oxaloacetic acid. Therefore, the first compound formed in the C4 cycle is a 4-carbon compound. Hence, it is called as the C4 cycle. And since it was discovered by 2 scientists, it is also called as Hatch and Slack cycle. So, it is also called as Hatch and Slack cycle. And there is one more name for it. It is also called as Kranz anatomy because it involves the mesophyll cells and the bundle sheath, wherein there is an anatomical modification wherein the bundle sheath forms a separate compartment like cell. So therefore, it is also called as the Kranz anatomy. So there are 3 names for it. Kranz anatomy. So, the C4 pathway is also called as Hatch and Slack cycle based on the 2 scientists who have discovered them, and also it is called as Kranz anatomy. So, here what did I tell? The first atmospheric carbon dioxide, it will be assimilated by phosphoenol pyruvate and then it will be converted or it will be fixed into a 4-carbon compound which is very stable, and that 4-carbon compound is oxaloacetic acid, and that will be transported. So they have written here C4 acid. It is nothing but the 4-carbon oxaloacetic acid, and that will be transported into the bundle sheath. Now in the bundle sheath here, decarboxylation will occur. So here what happens when decarboxylation? So what happens in the case of Kelvin cycle? Carboxylation, wherein carbon compounds get added, right? But in this case, one carbon dioxide molecule will get eliminated, that is it will get removed, that is decarboxylation. The removal of carbon dioxide will be done. The carbon dioxide that will be removed will be utilized for the Kelvin cycle. So next what happens here, the C3 acid will get formed, which is a 3-carbon compound. What is the C3 acid that is being formed here? It is nothing but the 3-phosphoglyceric acid, right? So here after decarboxylation, pyruvic acid will be formed, which is a 3-carbon compound. So pyruvic acid gets formed, which is a 3-carbon compound, and this pyruvic acid will be transported into the mesophyll cells, and here regeneration will occur. So what will get regenerated here? Again phosphoenol pyruvate will get regenerated, and this phosphoenol pyruvate, whenever favorable conditions will occur, or this carbon dioxide itself will be utilized, and when favorable conditions occur, it will again absorb the atmospheric carbon dioxide, and then the cycle will continue. So this is the C4 pathway. So here in C4 pathway, the thing that we understood is, first atmospheric carbon dioxide, that is will be absorbed by the mesophyll cells taking the help of a 3-carbon compound which is phosphoenol pyruvate, and this phosphoenol pyruvate will fix the carbon dioxide, wherein it will split the carbon dioxide into or convert the carbon dioxide into a 4-carbon compound which is oxaloacetic acid. That oxaloacetic acid will be transported into the bundle sheath cell which has been formed into a compartment through plasmodesmatal connections. So these are the plasmodesmatal channels or connections through which they get transported, and that C4 acid, which is nothing but a 4-carbon oxaloacetic acid, it will undergo decarboxylation, wherein the carbon dioxide molecule will be removed from that, and this carbon dioxide will be utilized later for the fixation by the Kelvin cycle, because for Kelvin cycle what is needed? Carbon dioxide is needed, but here under stress what happens? Limited amount of carbon dioxide is available for the plant. That is why in this process itself the decarboxylation of carbon dioxide removal will occur, and that carbon dioxide will be fixed by the Kelvin cycle, and meanwhile when the molecule of carbon dioxide is removed, what happens? It will get converted into the oxaloacetic acid will be converted into a 3-carbon compound, right? It was a 4-carbon compound. One carbon compound got removed, which will be utilized in the Kelvin cycle. So therefore, it will become a C3 compound, right? Which is a pyruvic acid. This pyruvic acid then gets transported back through the plasmodesmatal connection into this is pyruvic acid. It will move into the mesophyll cell and they get regenerated, wherein during the process of regeneration a phosphoenol pyruvate will be formed and therefore the cycle continues like this. So this is about the C4 pathway. So C4 pathway usually occurs in such plants which are subjected to a lot of stress, environmental stress. So this is about especially C4 pathway is seen in plants which survive in the desert conditions. So therefore, in desert conditions, we know that the plants almost most of the time in the daytime, the stomata is closed. So in such time, they make use of the carbon dioxide that is being present inside the mesophyll cell. That only will be made use of and it will follow a C4 pathway or Hatch and Slack cycle to complete this particular dark reaction. So this was about the C4 pathway. So next moving on to photorespiration. So here photorespiration. So light is being utilized wherein the RUBP, that is here RUBP is ribulose 1,5-biphosphate. So where did we find out about ribulose 1,5-biphosphate? In the Kelvin cycle. In the Kelvin cycle, what did ribulose 1,5-biphosphate do? So this ribulose 1,5-biphosphate is a 5-carbon compound, right? So this ribulose 1,5-biphosphate, it accepted the carbon dioxide from the atmosphere in the presence of an enzyme called as Rubisco, and it got converted into a 6-carbon compound. That 6-carbon compound got split into 2 molecules of 3-carbon compound, which was 3-phosphoglyceric acid, right? So this was what we observed in the Kelvin cycle. So here that was in C3 plants itself, right? So therefore it was also called, because the first compound that was formed was a 3-carbon compound which was 3-phosphoglyceric acid. It was also called as the C3 cycle. About all that we knew, where this is a kind of a dark reaction. We all knew about that. But sometimes in the C3 plants, what happens? In the atmosphere, oxygen will also be present, right? Is it only carbon dioxide that is there? There is also oxygen present. So sometimes what happens? Along with the carbon dioxide, some molecules of oxygen also enters and it will bind to the Rubisco enzyme. So therefore what happens? Carbon dioxide fixation will get decreased, right? So why? Because along with carbon dioxide, some molecules of oxygen also enters into the plant cells, and there it will go and fix with the enzyme, that is Rubisco enzyme, therefore reducing the carbon dioxide fixation. So we all know that carbon dioxide fixation or carboxylation is very, very important, because it is the first step in the Kelvin cycle wherein it converts the 5-carbon compound into a 6-carbon compound, and that 6-carbon compound will be split into 2 molecules of 3-carbon compound. So much process should occur. But because the oxygen will enter into the plant cell and it will bind to the Rubisco enzyme, the carbon dioxide fixation will get hindered. So here what happens? The RUBP, which is nothing but ribulose 1,5-biphosphate, which is a 5-carbon compound, instead of being converting into 2 molecules of phosphoglyceric acid, what happens? It binds with oxygen. Why it is binding with oxygen? Because here carbon dioxide is not getting fixed with the ribulose 1,5-biphosphate with the help of the enzyme Rubisco. Here what is happening? Because the oxygen is entering into the plant cells instead of the carbon dioxide, sometimes the oxygen will go and bind to the Rubisco enzyme. Therefore, instead of the carbon dioxide getting fixed instead of formation of phosphoglyceric acid, which is a 3-carbon compound, here the RUBP that is there, that is ribulose 1,5 biphosphate that is there, it will get converted, instead of being converting into 2 molecules of PGA. If carbon dioxide fixation had occurred, it would have converted into 2 molecules of 3-carbon compound, which is 2 molecules of 3-phosphoglyceric acid. But here what is getting fixed? Actually, oxygen is binding with Rubisco enzyme. So that is why here what happens, the RUBP will bind with oxygen and it will form 1 molecule of phosphoglycerate and 1 molecule of phosphoglycolate. So what happens? It will form 1 molecule of phosphoglycerate and 1 molecule of phosphoglycolate. So here, instead of carbon dioxide, oxygen is entering into the plant cell. Because oxygen is entering into the plant cell, what happens? The RUBP, instead of converting, so for you to easily understand, I'll tell about Kelvin cycle and then about photophosphorylation. In Kelvin cycle, what happens? That is in C3 cycle, carbon dioxide will enter, it will bind with the Rubisco enzyme, it will get converted into a 6-carbon compound. That 6-carbon compound will be split into 2 molecules of 3-carbon compound, which is 3-phosphoglyceric acid. So much should take place if carbon dioxide fixation is occurring. But here what happens, instead of carbon dioxide entering, oxygen will enter into the cells and that oxygen will bind with the Rubisco enzyme and it will get converted into 1 molecule of phosphoglycerate, 1 molecule of phosphoglycerate and 1 molecule of phosphoglycolate, which is a 2-carbon compound. And this pathway is called as photorespiration. So, instead of a 3-carbon compound being formed, because oxygen is binding with the Rubisco enzyme, instead of carbon dioxide being binding with Rubisco enzyme, what happens? Because your oxygen is binding, what happens? It will get converted into 1 molecule of phosphoglycolate and 1 molecule of phosphoglycerate. Both of these are 2-carbon compounds and this particular process is called as photorespiration. And in this particular photorespiratory pathway, there is no synthesis of sugar. Why no synthesis of sugar or glucose? Because if synthesis of sugar or glucose has to occur, then carboxylation has to occur, right? Or carbon fixation should occur. But here that is not taking place. Instead of that, what is happening? Oxygen is binding with Rubisco enzyme and being formed into 1 molecule of phosphoglycerate and 1 molecule of phosphoglycolate, both which are a 2-carbon compound. That is why here there is no synthesis of sugar, nor there is synthesis of any ATP. ATPs will also not get synthesized, sugars will also not get synthesized. But what happens in this case is there will be release of carbon dioxide with the utilization of ATP. Carbon dioxide gets released, that is decarboxylation will occur instead. So carbon dioxide will get released by making use of the adenosine triphosphate and in this particular pathway, that is photorespiratory pathway, there is no synthesis of ATP or NADPH. Whatever ATP was being available, that will be made use of and that carbon dioxide will get eliminated, that is decarboxylation will occur, but new synthesis of ATP nor NADPH will occur, nor they will synthesize any glucose. So this particular pathway occurs when plants sometimes take in oxygen or when oxygen binds to the Rubisco enzyme instead of carbon dioxide binding. So that particular kind of pathway is called as the photorespiratory pathway, or the process is called as photorespiration. So here photorespiration, that is how it occurs in the case of C3 plants. So in C4 plants, photorespiration will not occur. So photorespiration does not occur in the C4 plants. It is only seen in the C3 plants. Why photorespiration does not occur in the C4 plants? The reason is C4 plants have a mechanism that increases the concentration of carbon dioxide. At the enzyme site, right? At the enzyme site, how concentration of carbon dioxide increased? Because of decarboxylation process, right? If you remember the Hatch and Slack cycle, you would have understood about that. So here, the concentration of carbon dioxide will increase in the case of at the enzyme site of the C4 plant, right? So that is why C4 plants do not need photorespiration process, and this actually takes place, that is the increase in the carbon dioxide in the C4 plants actually takes place when the C4 acid that from the mesophyll cell. What was the C4 acid? The oxaloacetic acid that was formed in the mesophyll cell. If you remember, the carbon dioxide that was there in the mesophyll cell was utilized and it was broken down by PEP into a 4-carbon compound which was oxaloacetic acid, right? So that oxaloacetic acid that is formed, that is in the mesophyll cell, it gets broken down in the bundle sheath cells to release carbon dioxide. So what happened in the bundle sheath cell, Hatch and Slack cycle or the C4 pathway? The oxaloacetic acid got broken down into a pyruvic acid. During that process, decarboxylation is occurring there. That is, loss of carbon dioxide is occurring. So when there is the decarboxylation occurring, in the intercellular spaces of the mesophyll cells, the carbon dioxide concentration will increase. Right? Here, carbon dioxide is not being utilized. Decarboxylation occurs. But in C3 plants, carboxylation occurs. No decarboxylation. So, if all the carbon dioxide is being utilized, where will the carbon dioxide be available later on? No, right? So, that is why here in the case of C4 plants, here decarboxylation is occurring. Therefore, it will increase the concentration of the carbon dioxide molecules in the mesophyll cells, that is in the intracellular spaces of the cells. And when the concentration of carbon dioxide increases, the Rubisco will start to function. So, Rubisco will start to function. Why will Rubisco start to function? Because what is the function of the Rubisco enzyme? That is, it starts to function as a carboxylase minimizing the oxygenase activity. It will start to function wherein it will start to bind with the carbon dioxide compound and it will enter into the C4 pathway. So that is the difference here. And therefore, C4 plants, they lack photophosphorylation. Therefore, the productivity and the yields in C4 plants are quite better when compared to that of the C3 plants, and also these plants show higher tolerance to environmental stresses. So that is about the C3 and the C4 plants. So in C4 plants, because oxygen enters into the cells and it combines with the Rubisco enzyme, therefore what happens here? In the C4 plants, there is no production of the glucose and further process or the cycle will not continue. There is no production of glucose, neither there is production of the ATP nor the NADPH. Instead, they form a 2-carbon compound, 2 molecules of 2-carbon compound. We saw about that earlier. But in the case of C4 plants, what happens? Here photorespiration is not required. Why? Because even though if oxygen enters into the plant, but because of the process of decarboxylation occurring, already some amount of oxygen that is there inside the mesophyll cells will be utilized by the plant, and that in the process of decarboxylation, it will be again to the intracellular spaces, again carbon dioxide will be given back to the mesophyll cells, therefore increasing the concentration of carbon dioxide, and then therefore that carbon dioxide will be utilized in the C4 cycle or in the Kelvin cycle in the C3 plant itself, and therefore the plant will be able to survive under any stresses. That is why C3 plants cannot be grown in all environmental conditions, but C4 plants can survive both in the desert or extreme environmental condition as well as in the mesophic condition. So, or in the temperate and the tropical conditions. So, that is the difference between the photorespiration in the C4 and the C3 plants. So, in C4 plants, there is no photorespiration. What is the reason? Because of decarboxylation, photorespiration is not needed. But in C3 plants, because no decarboxylation is occurring, every time the cycle continues, if carbon fixation has to occur, it is not possible. At that time, photorespiration will take place. So, that is the difference between the photorespiration in the case of C3 plants and the C4 plants. So I hope you understood this session well, wherein we studied about the two important cycles under dark reaction. One was called as the C3 cycle or the Kelvin cycle, and the other one was the C4 cycle, which was also called as the Hatch and Slack cycle, and the Kranz anatomy. And also we studied what is the difference between what is the photorespiration, why photorespiration occurs in the C3 plants, whereas why photorespiration doesn't occur in the C4 plants. So I hope you understood the session well. In the coming session, we shall discuss about certain factors, that is intrinsic or internal factors and extrinsic or external factors that actually affect the rate of photosynthesis in plants. So there are various factors such as light, carbon dioxide concentration, and temperature, water. All these are the extrinsic factors, and when it comes to intrinsic factor, we shall know how the shape of leaf, position of leaf, size, number of cells, and chloroplast, chlorophyll pigment, all that plays an important role in the rate of photosynthesis. So all this we shall study in the coming session. So we shall meet again in the coming session. Thank you.